And now Tom Speth from the Office of Research and Development will moderate the webinar and introduce the speakers.
All right, thank you, Anna. Can you hear me?
Yes, I can.
Okay, great. Okay, so thank you very much for attending our webinar. This is a repeat of the November webinar, which was actually held on Halloween. And of course, we had some spooky internet interference. And so about half our attendees couldn't sign in, and it affected us too. And so we decided that in December, we would try our luck again and do a repeat of state acceptance of UV disinfection technology. So welcome to our webinar, and hopefully, because it's not Halloween, we should be okay in the technology realm. Okay, before we start, there's a webinar support phone number on the screen here if you're having problems. Hopefully we won't have them this time. Also, that recordings can be found on the link that they're up on the top. If you're interested in a recording of this webinar, you can find that at that website. We're very glad to offer a certificate of 1 continuing education contact hour will be offered for this webinar. So this is our standard disclaimer. Feel free to peruse it at your leisure. The requirements of the certificate of continuing education contact hour are listed here. You must be registered for the live webinar or be in a room with somebody who is. You must attend for the entire 60 minutes. If you're in a room with others, the names of people not logged in must be provided by the person who is, and send your names to the email address there. Or you can, if you didn't request the credit at registration, you can email the request to that same address or respond to the registration confirmation email. Unfortunately, we can't give out a certificate of completion for viewing the webinar recordings, only this live webinar. Okay, so last month we indicated that in November, okay, this one was in October because it was Halloween. The October one is being repeated now. In November, I mentioned that we have a Survey Monkey out there, and here's the link of the Survey Monkey. We want your opinion on what we can do better. So it's completely anonymous, so you don't have to say who you are, but we'd like to know how we're doing and how we might improve. We want to know the topics that you would like to be included in this series. Is there a topic that you think is really valuable for small systems? Let us know about that. Also, if you know about any communities or states that are possible test cases or somebody who would be an excellent speaker for that webinar, please let us know, and we'll try to do our best to incorporate that into this webinar series for next year. So we plan on doing our monthly webinars for next year also. So we value your feedback, and please take a few minutes to respond to that Survey Monkey. Okay, so let's get right into the talks. So our first talk is by Derek Loesch. Derek has 17 years of experience. He's been an environmental engineer with EPA's Office of Groundwater and Drinking Water since 2004. His initial experience was in developing regulatory determination for drinking water contaminants. Now most of his time is spent on implementation of the Unregulated Contaminants Monitoring Rule, or UCMR. More recently, he's been involved in projects initiated by EPA's Environmental Technology Innovation Clusters Program. And with that, I'll hand the podium to Derek.
Hello, everyone. I'm happy to be back again. Jeff and I are going to do our best to make this worth your while. It will be a repeat of, essentially a repeat of what we did back in October. So while I don't remember exactly what I said back then, if I happen to differ in any way from what I said back then, just choose the comment or the answer that suits you best. So the part of this project that I was lucky enough to get involved with came as a result of the formation of a drinking water cluster in the Cincinnati area. And shortly after they formed, their name is Confluence, they came together to sign a memorandum of cooperation. This was in early 2013. And the focus of this MOC was to jointly consider the development of water technologies in the area, with particular emphasis on reliability, operability, serviceability, and we thought the states, if they collaborated, could work towards some sort of harmonizing of the testing protocols that are required for each of these technologies. No specific, I believe, technologies were necessarily called out, but soon one surfaced that seemed to fit the bill. And that's where EPA started to get involved, because at the time, Jeff Adams was planning his research study. I believe you were in the planning phases at that point, on UV. And what he wanted to focus on with his study, with help from the water clusters and water technology team here at EPA, is UV drinking water disinfection, with an emphasis on inactivation of viruses and a more refined operation of medium pressure reactors. We had known at the time and thought we could address with a joint work group approach some of these issues that we know the regulatory, the state regulatory agencies have with UV installations. The rate of adoption of UV treatment for actual inactivation credit in the U.S. is pretty slow still, although it varies from state to state, I'm sure. And the reactor validation itself is a very costly and complex endeavor. So there's certainly some challenges there, and we hoped we could bring the states in our region together to try to start to address them. So just a few points about UV's performance, some of its attractions. We find that lots of states and water utilities are interested in considering non-chemical options for drinking water disinfection in particular. The increased safety that goes along with not having to handle the liquid chemical management is attractive, and also these particular systems are ripe for real-time monitoring and even automated monitoring in some situations, which requires less operator attention. They also can have very small footprints, which is a basic result of the fact that the residence time in these reactors is only a fraction of a second, so they don't have to be big reactors. They can often be retrofit in an existing building. And we've known that UV is quite effective at low doses for many microorganisms, Crypto and Giardia in particular, which is where it's usually pursued for some amount of regulatory disinfection credit. However, viruses have been trickier. They're more resistant, it appears, and that's one of the reasons it was a key of Jeff's research. So on slide 9, I am showing just a compilation of data from the literature on UV dose response for a variety of organisms. You can see that the Crypto Giardia organisms are inactivated. This is 4 log inactivation at doses less than 20 millijoules per centimeter squared. By the time you get to viruses, though, it creeps up over 20. And in the case of adenovirus, which is recognized as one of the most resistant viruses, you see the dose required for 4 log inactivation can get up over 100. A few regulatory considerations. This first table that I'm showing of dose requirements was published by the EPA when it revised the surface water treatment rule. And along with the publishing of this table, EPA started to produce and eventually published a UV disinfection guidance manual that was released in 2006. So when this table was published, it became kind of the target doses for each of the various microorganisms and the desired log inactivation credits that water systems were wishing to receive credit for. And the UV guidance manual was our best effort at trying to give guidance to those who were attempting to approach their drinking water regulatory bodies for credit. Now, we don't plan, the EPA isn't planning right now a formal update to these dose tables, so I think they're still an important part of, an important regulatory driver, let's say. Now, as I said before, adenovirus is quite a UV-resistant microorganism, and it's become the organism that's used to establish virus inactivation credit, kind of the target pathogen, which provides a conservative goal for inactivation of virus in validation testing, though it's not used directly, as Jeff will describe later in his presentation. It is kind of the target pathogen that we are usually talking about when it comes to viruses. We weren't able to address that in a lot of detail in the UV DGM virus inactivation because there wasn't enough, the science was lacking at the time. There wasn't enough consensus about disinfection of viruses in particular because they're difficult to study. Now, states have been kind of wrestling with the UV DGM ever since it was published. Some have made more progress than others, but EPA realized that it was, in some cases, a regulatory hurdle for those water systems who wanted to use UV. So we created this 3-state work group. The local states of Indiana, Kentucky, and Ohio each nominated representatives from their drinking water staff to meet in Cincinnati from time to time and begin a discussion about how these 3 states might come together on an agreed set of criteria or priorities for reviewing and approving UV technology in drinking water. We've hosted multiple meetings per year ever since 2014, and we've made a considerable amount of progress that I want to share with you. Some of the characteristics of the states in our region I want to summarize for you here on slide 13. We have Ohio has 4 very large surface water plants using UV. All have been awarded disinfection credit. One of them is not actively using it at the time, but may in the future. 2 more large surface water plants are currently seeking credit. And there's probably over 50 small groundwater plants, which is more pertinent to this, perhaps the small systems webinar series here that we're participating in. But it's interesting that none have applied for disinfection credit as of now. The story is kind of similar in Indiana. There's a number of surface water plants using UV, a slightly larger number of groundwater plants using UV, but only the 2 large surface water plants have been awarded disinfection credit. And we'll see if others are interested as we go on here. Kentucky has several plants using UV, and one system is now currently seeking approval. So they're right on the cusp of wrestling with this as a state in terms of awarding disinfection credit. Just to continue sharing some of the characteristics, we've got, like I said, the vast majority of PWSs using UV are surface water plants with medium pressure reactors, and they seem to be more likely to pursue disinfection credit. The small water systems are mostly groundwater plants using low pressure reactors, and they very rarely in our area seem to be pursuing disinfection credit. Now it's interesting, it leads you to wonder why water systems would use UV without an interest in pursuing disinfection credit for it. And some of the reasons that have been cited are that they just appreciate the extra level of public health protection. They believe they can reduce the number of microbial illness, stomach problems, and such for their customers. And it may allow them also, the technology, to remove specific chemical contaminants if they use it as part of an advanced oxidation process. So they're thinking of that possibility as well when they install these systems. Now, when we gathered and began discussing the process for UV review in our states, we realized there were two phases. One is the construction plans and specifications review phase, and we found that the states were fairly comfortable with that because it parallels very much other types of construction projects that are performed at water plants. But the UV reactor validation step was a lot more challenging, and that's what we chose to focus most of our energy on. There is, of course, also the monthly monitoring that's required for water systems that want to receive ongoing credit. Now, we found that both Indiana and Ohio, who have awarded disinfection credit, are relying largely on the UV disinfection guidance manual, and their customers or their water systems have used independent UV testing centers for validation and reporting. So all three states would like to see UV used efficiently and to its full potential. There's clear agreement on that, and they're willing to put in some effort, as we have done over the past couple of years, to make this a more viable option. They don't want the requirements to be onerous, but they certainly want them to be sufficient and protective of drinking water customers and, of course, defensible. The states believe the validation and monitoring requirements should be consistent regardless of water source type or water system size. And they realize the UV DGM is only guidance, but without industry consensus, they are not comfortable deviating too far from it because the knowledge that went into it is often a body of knowledge that the state regulating bodies themselves don't possess, and they're relying on the industry experts that went in, that participated in the development of the guidance manual. That's not to say that deviations from the UV DGM are unacceptable, but they're really hoping that the validation centers will explain any deviations and tell them why they occurred and what the repercussions are potentially for the validation testing. So when we started to discuss some of the considerations of these validation reports, Jeff Adams was really involved in leading a lot of these discussions because of his knowledge of the details of laboratory testing and full-scale testing. He explained to us the evolution in UV reactor test designs and analysis that have occurred over the years and are still occurring. Explained some of the challenges for validating UV for virus, and cited some of the also challenges and opportunities for crediting medium pressure reactors for inactivation at microbes at low wavelengths. These are, I think, very positive developments that the states want to become comfortable with. We also did a workshop over a couple of visits where we reviewed an actual UV validation report, and that allowed us as a group to define what we thought needed more clarification, where we could maybe add some insight that we could share with other state agencies that are interested in pursuing UV. Just a quick summary of the validation objectives so that you know what kind of questions, concerns we were wrestling with during our discussions on validation. We know that the goal of the validation testing is to determine the conditions under which a UV reactor delivers the validated dose. And that validated dose, which is determined through testing, must be greater than or equal to the required dose that I showed in the EPA's table. Now, the other part of the validation testing is to determine the operational set points that are used during reactor operation to confirm that you're delivering the validated dose. And like I said before, independent testing centers are usually the source of the ones who actually perform the testing and provide the reports for the regulatory bodies to consider when reviewing the technology. Now, the final result of all the testing is a dose delivery algorithm with a defined range of operation that is used once the UV reactor is installed. Here are some of the challenges. Number one, you can't measure the delivered dose in the reactor, at least not directly, as I understand it. We do measure UV intensity with sensors installed at certain points within the reactor, and we use that to as an input into the algorithm that defines the operation of the reactor. But it's really not feasible to measure the dose delivered to the microorganisms as they pass through this reactor at different trajectories all throughout, at different locations in the reactor. And every reactor is different. The hydraulics, lamp configuration, sensor locations, these all affect its performance. And finally, you can't perform testing with the actual pathogens. It generally poses an unwarranted health risk, and really analytically can be too difficult to attempt. So these things make it difficult when testing and evaluating the resulting data from these validation tests. Some of the solutions to those problems are we use these challenge microorganisms instead of the real thing. You'll hear Jeff talk about these more. MS2, B. pumilus, AD2, and T1 are some of the most commonly used challenge microorganisms. We do test the reactors at full scale to define the full operating envelope. And we do bench testing along with the full-scale tests so that you can deliver dose, or you can directly measure dose delivered to the microorganisms at the bench scale and use that information to back calculate the dose that you assume you are creating in the reactor itself. After that, you have to apply a variety of bias and uncertainty factors depending on how your study was designed. And this effectively raises what the required doses are in the EPA table to achieve the various levels of inactivation. Now, what we decided as a work group, the 3-state work group, is that we want to develop something along the lines of a UV toolkit to develop a package of documents that would aid state reviewers in evaluating UV technologies. It's not our intention to deviate from the UV DGM, but simply to provide a more narrow focus on the common concerns and priorities that our state reviewers think are important. By putting this out there, we hope to share with the industry what a better understanding of what the states in our region are looking for when they review UV treatment proposals. And we are going to focus, at least initially, on the current state of technology, which is primarily concerned with Cryptosporidium and Giardia, though we are interested and will discuss some of the evolving processes that are being used to hopefully make virus inactivation a more likely possibility for the future, at least credit for virus inactivation. Here's a quick outline of the framework for the UV toolkit. We're going to have a design review section, which basically addresses the way the water system has decided to install the UV treatment system in their plant. There will be a section on disinfection credit, which walks the state through determining what level of inactivation credit is appropriate. There will also be a section on operational requirements and a kind of framework that states can use to write summary letters that they can provide to their water system, so there's a common understanding of what's agreed to and what credit was issued. And we hope to make this toolkit publicly available in spring of 2018, so we're working hard on finalizing it right now so that we can get some review from our peers. Let me, before I turn it over to Jeff to see what he has been up to, I just wanted to thank some of the people who've helped us out here along the way. Sally has provided a lot of focus and even some funding for our work. My supervisor, Greg Carroll, has been very supportive. And of course, Mike Finn, who's in a sister office in Washington, D.C., has been very helpful. So with that, we'll introduce Jeff.
Okay. All right. Thank you very much, Derek. There's a couple of housekeeping duties I forgot to mention at the beginning of the webinar. First of all, if you missed the poll question, and if you have more than one person in your room, please type that number of people in your room in the questions box, which is in the dialog box off in your corner probably of your screen. That helps us document how many people are attending the webinar and helps justify the webinar's existence. So only do that if there's more than one person in your room, because we already know if you're logged in that there's at least one person in your room. But if there's more than one, please do that. Also, in that dialog box, the question box, feel free while the speaker is presenting to type in your questions ahead of time. It helps us organize the questions for after the second speaker, and then we can get started with the questions right away. So again, feel free to put those questions in the box. So with that, I'll introduce Jeff Adams, who's our second speaker from our Halloween redo. So Jeff is an environmental engineer with EPA's Office of Research and Development Water Systems Division. He has conducted studies evaluating performance and sustainability of water treatment and monitoring technologies, including filtration processes, membrane separation, adsorptive media, UV and disinfection processes, and advanced oxidation technologies. Jeff has managed, authored, and co-authored numerous technical articles and has been active with the American Water Works Association and the Water Research Foundation. Received an MS and a BS in civil/environmental engineering from the University of Cincinnati. And with that, I'll hand off the podium to Jeff.
Thanks, Tom. So as Derek mentioned, over the last couple of years, EPA's Water System Division, which is what I'm in, has been conducting bench and pilot UV inactivation studies for drinking water with some emphasis on virus applications. In these studies, we have been evaluating alternative testing and analysis approaches compared to traditional UV DGM procedures. We conducted UV reactor studies for about 200 test conditions for both low pressure and medium pressure, using several test microbes, as Derek kind of pointed out. Medium pressure studies included also UV lamp monitoring, accounting for both low and high wavelength UV sensor measurements, particularly to support adenovirus crediting. And we incorporated the lessons learned from our EPA studies, along with recent industry experiences in a report to provide recommendations for future UV validation. So the UV DGM documentation is over a decade old now. As Derek indicated, EPA is not currently planning on a formal update to the UV DGM or the UV dose tables. So a driver for our studies has been to try to address a number of the weaknesses or drawbacks with the UV DGM realized over the years and address evolving UV challenges. So some of the UV DGM issues of concern include, or listed here, weaknesses with both the set point and the calculated dose approaches, action spectra correction factor determination and implementation with polychromatic systems, basically medium pressure systems, microbe QA/QC issues, statistical and uncertainty bias adjustment factors, also there was a lack of UV validation protocol for virus disinfection in the UV-DGM, and this would be particularly important for the application of UV under the groundwater rule. Also a need to assess new calculated dose algorithms with better accounting for UVT on dose distributions, microbe sensitivity, and full germicidal lamp output effects on inactivation, and assess the implications of using high dose microbes like Aspergillus brasiliensis and B. pumilus for predicting adenovirus inactivation. So let's unpack a few of these issues that I've talked about. The UV-DGM and O-norm set point approach has been shown that UV underdosing as much as 15 to 25% can occur with some validations due to the lack of intermediate UVT and lamp output combination conditions tested. So the two typical conditions tested are, 100% lamp output with lowered UVT versus high UVT and lowered lamp output. And this may not identify the low RED set point condition. So I've got two examples here that I show. In the upper graph validation, the set points bounding the blue validated box may actually be more stringent, shrinking the validated range. But we really don't know because our other test conditions were not assessed. So these points down here, I assume my cursor's working, yeah. These points down here could actually be a little bit higher, but we don't know because we only assess the two extreme conditions. In the lower graph here, is a validation using a calculated dose approach, and that shows the governing RED conditions are intermediate UVT, that is 80 and 90% with intermediate lamp output conditions, not the two set point extreme conditions. So this has been seen in a number of real validations over the years, and we're just beginning to address some of these issues now. So in our studies and report recommendations, we present a hybrid calculated dose method that captures the conservative conditions that you want to capture, but still provides an approach that does not require online UVT monitoring during reactor operations for small systems. So in a calculated dose validation approach, the experimental data is fitted to an algorithm like shown here that can be programmed into a UV reactor PLC, incorporating the parameters monitored at a water treatment plant for UV operations such as flow rate, UVT, and lamp output. So in our EPA studies, we employed a calculated dose method, but unlike the traditional UV-DGM algorithm, in our work, we have explored algorithms like shown here that incorporate the sensitivity of the test microbe, that's this DL factor here, which is dose per log inactivation, to assess and also assess the scalability of lamp output and flow rate, and which better defines the effective UVT on dose distributions. So, So during reactor validation, challenge testing is conducted with numerous conditions with one or more microbial surrogates. With the concept of microbial bracketing, the log inactivation algorithm is developed from validation testing with two or more test microbes having UV dose responses bracketing the target pathogen. And generally, it is impractical to test with crypto and Giardia, and for the most part, adenovirus. Bracketing assumes interpolation of microbe sensitivity for defining the reactor operating envelope. So, for example, if adenovirus is the target application, which in this example, you bracket the lower UV resistant end with MS2 and the more UV resistant end with B. pumilus or something like Aspergillus brasiliensis, in this case, B. pumilus. However, in our testing, we did do challenge testing with adenovirus type 2 to assess measured results in contrast to predictions with other test microbes as sort of a proof of concept. In our alternative approaches, one could use a bracketing test scheme like this, and we did that in our studies, but one could also predict a target pathogen using a single test microbe data set, which we also assessed using the new alternative approaches that I'll be talking about. So some of the slides will get a little bit complicated. Try to kind of break this down a little bit. But unlike the traditional UV-DGM approaches, in our studies we employed a concept with a combined variable, incorporating relative lamp output. And if you look on the x-axis, you can see the combined variable. S over S naught is relative lamp output divided by flow, Q, and microbe sensitivity. Again, DL is the dose per log inactivation from a dose response curve, or from your EPA dose tables if you're looking at a target pathogen. And again, this is shown in the x-axis, measured log inactivation on the Y. So the combined variable implies that UV dose distributions within a UV reactor scale proportional to relative lamp output and inversely with flow rate. This has been shown with years of CFD modeling and results from over 50 real UV reactor validations. So at a particular UVT, and we show here 70%, around 90, 98%, microbes with different sensitivities tend to correlate. So at a fixed UVT, log inactivation of differing microbes occurs at a similar value of the combined variable, that S over S naught QDL on the x-axis, which reflects scalability with lamp output and flow and sensitivity of the microbe. Again, this has been observed with numerous real reactor validations and modeling. So what this is it provides a means to assess the prediction of a UV inactivation performance of one target microbe from the validation data set of another test microbe, applying certain constraints and bounds. Another characteristic here is, you see graphically, is that the UVT correlations tend to show more curvature at lower UVTs and wider dose distributions. And this effect is captured using the algorithm that we recommend in our report. And again, why there's one of the weaknesses with the algorithm you use in the UV-DGM is it does not take into account a better approach for taking the dose distribution effect based on UVT. So in the application of limits and bounds being discussed, what we have here is some of the parameters. In applying the algorithm, the combined variable is limited to a tested validated range of values based on your test matrix. However, some of the individual parameters may scale outside of the tested range. What that means is that S over S naught QDL, this in the yellow parentheses here, that combined variable, you set that to the validated range of your test matrix. But some of those variables within this can vary when you do your analysis of comparing one microbe to another microbe. However, in most cases, we recommend that the flow rate in the UVT is limited to the tested range. So the implication is that systems could be validated with MS2 for the prediction of adenovirus inactivation. However, a second microbe data set like T1 is recommended during validation as a proof of scalability with the combined variable concept for each specific UV reactor. So in the example below is a T1 data set that's used alone for predicting MS2 in the Aspergillus brasiliensis with good correlation, even though the T1 UV sensitivity is significantly less than those other microbes being predicted. In the case of T1 compared to the Aspergillus, it's an order of magnitude. So just like the T1 prediction assessment, here's an example. We want to show how well MS2 microbes predict adenovirus inactivation performance. So shown here is measured inactivation versus the algorithm predicted values using MS2 data set alone. And MS2 alone provides a favorable comparable slope and conservative predictions of adenovirus performance. Now we do observe experimental noise with both adenovirus and B. pumilus microbes as expected due to variability of the microbial assays. We'll show a little bit some examples of those that variability later in some later slides. Generally, MS2 provides better adenovirus predictions than using B. pumilus alone or bracketing MS2 and B. pumilus. So in this kind of analysis using the combined variable, there's certain proofs that are recommended. Validation reports, as I mentioned, should include a minimum of 2 validation surrogates for example, MS2 and T1. Now, we recommend MS2 and T1 because they have low microbial variability issues as compared to other microbes, particularly the Pumilus and adenovirus, which has a lot of variability. So proofs must be conducted to show that the log inactivation models developed from a less UV resistant microbe like T1 can predict the performance associated with the more resistant microbe MS2 and vice versa. Similar proofs for models developed with high and low lamp output data should also be included. And if a reactor has a different set of lamp on-off combinations, the range of those configurations should be tested. In terms of developing your algorithm, the calculated RAD requirements are determined from your EPA dose tables, just like a traditional UV-DGM approach. And then the validated RADs are determined, as Derek had indicated, after adjustments based on certainty of validation calculation. This takes into account the uncertainties of sensors, dose response, and inherent data interpolation uncertainties. But the procedures that we present in our report are even more simplified than those in the UV-DGM guidance. The examples here are just, again, some other examples. I'm not going to go into detail, but again, showing MS2 and T1 predicting the significantly more UV-resistant Pumilus with good correlations is sort of an adenovirus dose range type of analysis. So kind of reiterating what some of the attributes are for this combined variable approach, you can see that basically a combined variable approach can be used to directly predict crypto and Giardia and adeno inactivation and generate an RAD algorithm from a single test microbe. This eliminates the RAD bias and simplifies action spectrum correction factors and makes the validation factor analysis even easier than what's in the UV-DGM. It eliminates RAD because basically you are incorporating the true RAD bias in your algorithm development. In comparing predictions, the other thing is that we also recommend comparing predictions from two test microbes, as I mentioned before, to offer a proof of application for a specific UV reactor. This also levels the playing field in terms of what manufacturers do in terms of this kind of analysis, because UV-DGM RAD bias factors tend to favor the selection of UV systems with wide dose distribution, but there's no bias of reactor type under the combined variable approach. So the validation approach for medium pressure systems has additional challenges, and we need to consider action spectra issues. So action spectra development has been addressed by a recent Water Research Foundation study, 4376. Action spectra correction factors account for differences in microbe sensitivity between a target pathogen and surrogates across the UV germicidal range from 200 to 300 nanometers. For example, MS2 is more sensitive to UV than crypto in the low wave interval, so validation should be adjusted to credit properly. Adenovirus, which we show here, is very sensitive to UV in the low wave, and we want to be compared to MS2, and we want to be able to take into account that sensitivity in our crediting for UV with adenovirus. So the Water Research Foundation report provides action spectra for several pathogens and surrogates used in validation, and we use that data in our EPA studies. So one of the take-home messages from the report is if there is no monitoring of low wavelengths, no disinfection credit should be given for the low wave interval contributions to inactivation. Now, a typical UV sensor focuses measurement on UV intensity in the 240 to 300 range. So unfortunately, the lower UV wavelengths are not measured below 240. the concern is that lamp degradation, quartz sleeve fouling, and UV absorbance can all vary at different rates in the low wave interval versus the high wave interval. So it is important to monitor the performance across the relevant intervals to get disinfection credit. So in our study, we assessed the first of its kind low wavelength UV intensity sensor for reactor validation and operation. However, an alternative to a dedicated low wave sensor, you could use a customized spectroradiometer to take measurements taken at particular intervals each day. And we assessed both of these approaches. So our concept was to monitor both high wave interval and low wave interval UV intensity during testing, as well as taking UVT measurements from both of those intervals. So a UVT at 254 nanometers and one at 220 nanometers. Validation included multiple test conditions for both maximizing and minimizing low wave UV contributions. We did this by using synthetic sleeves versus 219 quartz sleeves and two different UV absorbers. Calculated RED algorithms associated with the low and the high wave inactivation contributions were developed, and then a final overall RED model is determined. So even though the equation looks a little bit scary there, it's basically just a repeat of the low pressure equation that I showed earlier, one accounting for the low wave contributions, one for the high wave contributions. Validated REDs are then, determined or adjusted then based on uncertainty and validation factor. Basically, what this does, as Derek indicated, is that it essentially increases the required dose above the EPA table values to meet your target inactivation credit. So, for example, for 4-log inactivation, EPA requires 186 millijoules per centimeter squared, but after your uncertainty and bias factors, the required validated dose might be more in the range of a little over 200 millijoules per centimeter squared. So I just have a few other examples here to kind of make some points of interest. In our work, we rely a lot on MS2 because it exhibits consistent dose response. So over several days, the dose response is very close to each other, making it very desirable to work with in UV validation. In the graph below is the medium pressure algorithm results with the high and the low wavelength sensors and UVA measurements mapping the MS2 data, and it does it very well using the algorithm that we present in the report. I show adenovirus here because I want to show what kind of a pain in the butt it is to work with. The problem with working with adenovirus itself is that the dose response shows a lot of variability from day to day, and from even within the same stock solutions. The medium pressure algorithm using only the adenovirus data is shown in the graph below, and it shows a significant pattern of microbial variability resulting in experimental noise. Again, this makes adenovirus a poor test microbe. The other big issue is producing the stock solutions and performing the assays are extremely difficult and very expensive. It took us over a year to be able to propagate enough adenovirus at high enough titers to be able to do our work. Very difficult to work with. This is a look at B. pulmonis in the work that we did with B. pulmonis, and that this assessment shows that the dose response, again, is similar to adenovirus, so that you have, from day to day, you have significant variability in dose response, and even within a single day, and you're doing your samples with your reactor, there's a lot of variability in the triplicate samples that we get. We did assays with 2 different labs, and again, there's a lot of variability that we see. So we did a round robin with 4 different labs actually, and all of them showed extreme variability. So predictions, as you can see in the lower graph, show less favorable slopes and more data spread than using MS2 predictions. So using B. pulmonis alone as a single predictive test microbe may yield more uncertainty and appears to be less favorable than MS2 as a single microbe. This is a look at the medium pressure algorithm, again, using only MS2 data, predicting B. pulmonis. And it does it fairly well despite the microbial variability and adenovirus conservatively. So the MS2 does, again, like we saw in the low pressure example, predict the adenovirus conservatively with favorable predictive slopes. Also, the MS2 alone provided as good or better inactivation predictions of adenovirus than bracketing with both MS2 and B. pulmonis. So our draft report is anticipated to be in final in 2018. Again, to kind of reiterate a couple of things, this is not formal Office of Water guidance. It's not intended to replace the UVDGM. So there's a number of procedures in the UVDGM that are still very relevant, even though there are other procedures that we provide clarification in this report. And the intent is that you would follow these updated recommendations based on our report. So traditional UVDGM calculated dose and set point validation approaches have weaknesses, and the alternative approaches that we presented in our report for validating UV systems offers more direct prediction of pathogen inactivation from a single test microbe, offering also the proofs that you should follow during validation and the production of your validation report. So the report is undergoing stakeholder review with assistance from a number of UV industry volunteers. We have those that are affiliated with the IUVA, AWWA, Water Research Foundation. We have a number of folks associated with state regulatory agencies to get their perspective. So the kinds of things that are addressed in this report are, we present 4 different variant approaches for validation based on this combined variable approach. Some that incorporate the use of online UVT monitoring and some that don't. There's a discussion of test plans, data analysis procedures, the alternative algorithms to the UVDGM that we present, a discussion of what validated limits and bounds are for each of those applications. And very importantly, for the state regulatory reviewers, we present recommended contents for validation reports and checklists that we feel should be applied by manufacturers in doing their validation, so that we can get some more standardization in the production of validation reports. It also addresses, as I indicated, modifications to the UVDGM approaches that should be considered. We present microbial methods. We have updated QA/QC bounds that we present in the report. And then there's a whole section on the rationale and background of these new alternative approaches, and then the demonstration and the results of our demonstration studies that we conducted under our EPA work. So what's sort of the current status? A draft report is undergoing stakeholder review, as I mentioned. The purpose, excuse me, the purpose of the review group is to help define limitations and bounds for these alternative procedures that are being considered, and offer suggestions to add clarity to the contents of the report. I would say that the current report framework and language is likely to serve the UV consulting, testing, and academic community very well, as it presents significant technical details on testing and analysis approaches and UVDGM updates. I would say, unfortunately, one of the drawbacks is the recommended procedures and rationale are a bit complicated, in communicating this content to state regulatory permit managers and reviewers is a bit of a challenge. So one of the things that we would like to do is try to be able to produce a separate summary and toolkit document. Derek talked about one phase of the toolkit on behalf of the regulatory reviewer perspective, and then also from the manufacturing standpoint in ours. But we would like to be able to produce this sort of toolkit document that would help serve that regulatory and water utility community. And yes, this is a bit of an uphill challenge because EPA is facing a bit of limited resources for support of this effort and many other efforts. But we are continuing to explore with our current partners and collaborators to be able to meet this goal. a couple of things. What are some of the things that would be helpful both in this upcoming EPA report and a separate toolkit document, if possible, is one, be able to present more clarity for the specification of the types of recommended data, proofs, and content that we should expect to find in validation reports. That is, really would like to strive more for standardization in this process. We don't want to be overly prescriptive, but more standardization would be helpful for the regulatory community. I would say that, and as Derek talked about, his whole effort with the regulatory group from the Ohio, Indiana, and Kentucky, that continues to be ongoing. Beyond this report, we hope to continue to engage the state regulatory offices and industry work groups to improve the overall process. For example, one activity is the EPA is engaged with NSF International to collaborate in defining their new standard 420 for UV under their public water treatment certification program. Our new protocols will likely influence what ends up in the standard 420, which will probably be a more cookbook version of an approach for specific applications in which they want to offer certification. So just a couple of brief acknowledgments. Funding for this effort was provided by the Water Technology Innovation Cluster Program and also the Water Systems Division that I belong to. The EPA project team included both Office of Research and Development, Office of Water collaborators. I want to say special thanks and appreciation to those who provided valuable microbiology support during this testing. And then contract technical support included a number of UV experts, some of which were also involved in the original UVDGM development effort. So with that, maybe we can move on to some questions.
Okay, thank you very much, Jeff, and thank you very much, Derek. We're moving on to the questions section. Before we do, the couple things to note is if you missed the poll question and you haven't already put the number of people in your room, if it's more than one, please do that in the questions box. Also, we've received some questions today, but if you have a question, feel free to put it in the box. We'll get to it. We did this webinar on Halloween, and we had about 750 or so people sign into that one, but there was this nationwide type of thing going on with the internet, and we knew we missed a lot of people. And then this one, we're somewhere around 350 people. So that brings us to kind of what we expect, because usually we get over 1,000 people, slightly over 1,000 people tuning into these webinars. So thank you that you were able to pop into this webinar. So we might not have quite as many questions this time because we've done this before, but if you have any questions, feel free to throw it in there. So again, I thank Derek and Jeff for these excellent presentations. If you have to leave us, because I know some people have to leave on the top of the hour, January 30th, we're going to do a webinar on chlorinated thiurates, like dichlor and trichlor. We know that this is something small utilities put in to maintain a chlorine residual. It's a little more, keeps a chlorine residual a little more stable, but there's some question about their disinfection efficacy or capability. So we're going to cover that on January 30th. So please tune in for that webinar.